welcome to week 5. This week we are going to talk about gene regulation. So we're going to take some notes, go ahead and get a pen and a piece of paper, and let's get started. Our topic is gene regulation and gene expression. So you might wonder, what is gene regulation? Gene regulation is controlling the way your genes are used. And the way that they're used is referred to as gene expression. So you're regulating how you're using them. How you're using them is called expression. What this really means is, are your genes turned on or off? If your gene is on, that means that you're going to be making a protein. And if your gene is off, then there's no protein. We know it relates to protein because each gene is the genetic code to make one protein. So our gene regulation, we're going to be turning genes on and off, which means we'll be making some proteins and not others, which is actually pretty cool, right? Because if I have a regular cell right here, this is a eukaryotic cell, so I have a nucleus. Inside my nucleus, I've got DNA. So cool. All of the DNA inside my cell we call the genome. The genome of my cells is the same in all of my different types of cells. So genome is all my DNA. That means that the genome inside of our 4 types of tissues that make up humans is the same in all of our cells. So the genome inside of a cell that is made for movement, this is what a muscle cell looks like, is the same as the genome inside of a cell that we use for thinking. So this is a neuron cell. We use these for thinking. Remember, everyone is a fantastic artist. The genome inside of something like the cutest cell in your body, which is an adipose cell. Here's your nucleus. And then the cool thing about an adipose cell is almost all of the inside is filled with lipids. If you haven't heard the word adipose before, adipose is fat. So these are the fat cells in your body. When you gain and lose weight, it's this lipid droplet inside that shrinks or swells. Or one of your skin cells. We call those epithelia. They kind of look like fried eggs from the side. Whoop. See the fried egg? And then this is where your nucleus is. These are the 4 types of tissues that make up a human, and they all have the same DNA, but they're totally different. And the reason that the skin cells are not the same as the fat cells are not the same as the neuron cells is because of gene regulation. We're only turning on the genes that we need to make muscle protein in the muscle cell. The proteins that are used in muscle are called actin and myosin. That's what makes a muscle cell able to contract. Contract means movement, so I'm using my muscles to do all these little motions here. If I had a bunch of actin and myosin in another cell, it could function like a muscle. My adipose gene that allows it to store all these lipids makes it different than my neuron gene, which allow it to move ions around. That's what gives it an electrical impulse. So all of these different types of cells have the same genetic information, but different genes are turned on. So I have a bunch of sodium-potassium pumps in my ion. I've got all this hydrocarbon in my adipose cells. And so changing what part of the genome you're using changes what the cell does. So the function of the cell is determined by what parts of the genome you're using. So how do we turn those genes on and off then? Gene regulation. I'm going to add my heading back. And it turns out that I can turn my genes on and off in different ways, depending whether or not I'm a prokaryote or a eukaryote. So which are you? Are you a prokaryote or a eukaryote? And hopefully you answered you're a eukaryote. You know you're a eukaryote because you have a nucleus. Oh, spell nuclei right. Nuclei in your cells. Prokaryotes don't. You know that you're a eukaryote because you have organelles. Like mitochondria, or if you were a plant, you'd have chloroplasts. Super cool, but you already know this because we covered this last term. What we really want to talk about is the idea that gene regulation is different in prokaryotes and eukaryotes. Prokaryotes have a lot simpler mechanisms for gene regulation. In a prokaryote, you're going to do transcription and translation at the same time. Remember, transcription is making mRNA from the information in DNA, and translation is taking that mRNA and making a protein. So prokaryotes do this at the same time, which means if I wanted to control translation, I would need to control transcription. So most of gene regulation in a prokaryote, most gene regulation, is therefore transcriptional. What that means is no mRNA, no protein. If I wanted to make more, I'd make more mRNA. If I want to make less, I make less. But all I have to do is control transcription, and I can control what kind of proteins I'm making. It's a relatively simple way to do gene regulation. Eukaryotes, on the other hand, we're way more complex. We have a lot more going on. So let's think about eukaryotic gene regulation. Eukaryotic gene regulation. Turns out there's 5 ways to do it. So the first one is going to be just like prokaryotes, we can do it transcriptionally. When we're doing transcriptional gene regulation, what we're really looking at is how much mRNA is made. If I make a lot, I can make more protein. If I make a little, I'll make less protein. So pretty awesome, similar to the prokaryotes, except then it gets way more complicated because we don't make transcription and translation happen at the same time. Instead, we make them separate processes. So now I have another way that I could regulate my gene expression, and that's via post-transcriptional control. So post means after. And you know transcription is the process of making mRNA. So post-transcriptional means we're going to regulate how the mRNA is modified. There's a couple things that we do to mRNA to make it mature. Remember, we need to add a cap, 5' cap. We need to add a poly A tail. We need to splice it. So remove the introns. All of these mechanisms can be modified so that our mRNA is different. Let me give you an example. If I had a piece of mRNA here, and it's got a couple introns, if I were to modify this mRNA to make it mature mRNA and I removed all of the introns, and then another time I wanted to modify it to make it mature, but I only removed this middle intron, or another time I removed it, but then I swapped the position of the exons, each of those would result in a different mRNA molecule. When that mRNA molecule went over to the ribosome to be translated, I would then produce different proteins. So how I modify my mRNA can affect what proteins are being produced, which is a form of gene regulation. Because remember, our genes are what we use to make proteins. So a lot of things that can go on in post-transcriptional gene regulation. The third way that we can modify our genes is via translational. So transcriptional, post-transcriptional, translational regulation. So translational regulation will mostly be how are we using the mRNA. So how much mRNA is made, how your mRNA is modified, and then how are we using the mRNA? Are we using the whole mRNA molecule? Are we using part of the mRNA molecule? Is the mRNA molecule short-lived? Does it only get translated a couple of times and then it degrades? All of that would be involved in translational gene regulation. Just like we had a post-transcriptional, our fourth option is going to be post-translational. In post-translational gene regulation, you're going to be looking at how the protein is being used, because you know that you made a protein because you did translation. We can mark some of our proteins to be short-lived. They can degrade really quickly. So if I want it to be a short-lived gene expression, that would be post-translational. Some proteins are longer-lived, so its activity level is a part of this. And how long it lasts. And then the last way that eukaryotes can regulate their genes is with epigenetic gene regulation. So epigenetic means above or upon, that's what epi means. If you want another use of epi, an epiphyte is a plant that lives on another plant, like mosses live on trees or ferns live on branches. So epi means upon or above. So an epigenetic gene regulation is going to be regulation of your genes without changing your genome, which is cool. We haven't changed our genome in any of these situations, but these are often environmental factors. So the way this works is we want to affect how our genes are accessed. So how our genes are accessed. If I have a epigenetic regulator that I want to be really tightly controlled, I will add methyl groups to my histone proteins. Remember, a histone is a protein, and then DNA wraps around the histone protein. So that's it wrapping around. This is DNA. This is the histone. Together we call this chromatin. The mixture of DNA and histone proteins is called chromatins. If I wrap my DNA really tightly around this histone protein, it's harder to access the pieces of the DNA that code for protein. So I'm literally like tying it tight around this protein, which makes it harder to get to. If it's harder to get to, I'm going to decrease the transcription rate, I'm going to decrease the translation rate, and that's going to make my overall gene expression decrease. The way that I wrap it tighter is I put these little tags on it called methyl groups. So methyl decreases your gene activity because it wraps it tighter. There's another way to do it where I can make it more accessible. Then I'd have my histone protein. I've got my DNA wrapping around it. So it's being wrapped around that protein. Here's my DNA. If I added an acetyl group, that makes this DNA loosen up. So it becomes less tightly wound around my histone protein. Since it's looser, it's easier for your cell to access it. So acetyl groups increase gene activity. There's a really cool, fun thing we're going to do in lab next week where you're going to affect the methylation or the acetylation of different genes in rats. So this is a pretty cool thing that we've done experiments with. But we're changing your ability to access the genes. So it is epigenetic gene regulation. How you access your genes controls how often you can do transcription, how often you can do translation, and both of those affect what kind of proteins you're making, and those proteins determine the function, the shape, and the activity of your cell. So gene regulation, how our genes are being used. Okay, hopefully that helped explain how gene regulation, gene expression, and transcriptional, translational, and epigenetic vocab are all related. If you have any questions, send me an email, and I hope you have a great rest of your day.